Welcome to our final lecture in the ruminant digestion module, week 6, lecture 1, titled Microbial Fermentation of Fat and Avian Digestion. In our previous sessions, we examined microbial fermentation of carbohydrate and proteins, understanding how these nutrients are broken down by ruminal microbes for absorption. Today, we will complete this module by exploring the unique processes involved in fermenting fats within the ruminant digestive system. Additionally, we will introduce the fundamentals of avian digestion, comparing it to ruminant and non-ruminant systems to highlight both similarities and difference. This lecture will provide a well-rounded understanding of digestive strategies across species. Let's conclude this module with a comprehensive look at these final topics. Dietary lipids. Dietary lipids is of either in form of structural lipids, that is in the leaves, or in oil seeds, that is a storage form of lipids. So either it is a structural lipid or it is a storage lipid. The structural lipids, if you see in the leaves, it has less than 50% are of free fatty acids. That is, majority is of phospholipid in nature with palmitic, linoleic and linolenic acid. Okay. Whereas coming to the storage lipid, that is in oil seeds, around 65 to 80% are triglycerides in nature with palmitic, oleic and linoleic. Now, the dietary lipids is an unesterified fatty acids. You know, whenever you see the fatty acids, there are two types. One is saturated fatty acids, another one is unsaturated fatty acids. You might have come across these terminologies. What is the saturated fatty acids? The saturated fatty acids are nothing but these are the fatty acids which contain single chain of carbon atom with no double bond. Okay. Whereas the unsaturated fatty acids, you can say here you may have either one or more double bond. Okay. So examples for saturated fatty acids, you can say animal fat, then coconut oil, etc. Whereas the unsaturated fatty acids, you can say the plant and vegetable oil, sunflower oil, fish oil. These are all the example of the unsaturated fatty acids. Now let us come to the business. Dietary lipids, that is unesterified fatty acids, which is hydrolyzed by the ruminal microbes. And these ruminal microbes are releasing the different enzymes. We have seen even in the fermentation of carbohydrate as well as the microbial fermentation in the proteins also, where these enzymes are released by the microbes. Similarly, here also these microbes releases an enzyme lipases. So these lipases will act on the dietary lipids and hydrolyze them, yields unsaturated fatty acids such as oleic, linoleic and linolenic. Then these unsaturated fatty acids in turn accepts the hydrogen ion and convert most of them into the stearic acid or cis trans monoenoic acid and cis trans dienoic acid, which is an end product of the lipid digestion. Among this end product, the stearic acid is an important fatty acid because it is a predominant acid of storage as in the ruminant as a fat depot. That is the most important aspect of the stearic acid. The fat which is stored in the ruminant is majority is of stearic acid in nature. Okay, that is very important. Then the ruminal fauna or the bacteria, especially you can say, the synthesizes the odd chain fatty acids from propionate and branch chain fatty acids from valine, leucine and isoleucine. Even in the protein, we have seen that the branched volatile fatty acids are produced by utilizing this valine, leucine and isoleucine, right? The organic acids. Then the next important is that phytal. Phytal is present in the chlorophyll of the leaves, where the rumen bacteria acts on the phytal, and it undergoes two important biological processes such as oxidation and the hydrogenation. This oxidation and hydrogenation of this phytal will yield the phytanic acid. Okay, so these are some of the important aspects of the dietary lipid digestion in the ruminants. Now, if the ruminant diet contains more lipid, that means more than 5% of its total, then what will happen? It leads to some adverse effects on the certain things are there that we'll see now. First is that palatability. What is that palatability? Palatability means it is the likability of any feed. Okay, palatable, right? Something. So if something is not palatable, if you don't relish, if you don't like, then you may not consume. Like sometimes if you like only one type of chocolate, like example, you can take 5-star or dairy milk. If you like that chocolate, you will take more. But on the other hand, if someone gives you the Kit Kat or something else, then that means you may not like it or relish it, right? The same way, even if someone provides you with a sweet or someone is providing a bitter gourd item, then obviously we'll go with the sweet, because that palatability is the important one. So if any diet or the feed substance which is having a more than 5% of fat in it, then the animal, it may not be palatable to the animal. That is first important adverse effect of higher lipid concentration in the diet, dry matter content. The next is that cellulolytic activity. How the cellulolytic activity get decreased because of this high, it is also another important aspect, the adverse effect of the high dry matter as lipid, that means more than 5%. Then the food appetite, I will come to this cellulolytic bacteria soon, right? The food appetite also decreases, and then stomach motility get decreased. How? Because of the negative feedback mechanism of cholecystokinin. Now, what this cholecystokinin does? It causes the digestion of the lipids and enhances the absorption process of the lipids. So, this cholecystokinin releases when there is a lipid in the duodenum, because it has to be absorbed, right, by the lacteals. Now, if the CCK won't does this negative feedback mechanism to the stomach, then what will happen? As a result of increased motility, more and more the chyme will enter from the stomach to the duodenum part. So as a result, without having the maximum absorption, this chyme will move to the next compartment of the next part of the small intestine with lesser absorption rate. So in order to enhance the increased absorption rate in the duodenum part, the CCK produces the negative effect on the stomach motility. As a result, less ingesta is sent to the next compartment, that is the duodenum, until unless it is absorbed. Okay, the clearance rate, you can say another way. Then the shelf life of the concentrates also as an important in the diet, because if you keep anything which is having a high concentration, then that will lead to the rancidity flavor. This flavor may not liked by many animals. As a result, one is that the palatability decreases, and the other way is that the shelf life of the concentrates also decreases or adversely affects. So these are some of the ways where when you increase the concentration, the dry matter of the lipid more than 5% in a diet, then these are the adverse effects you can see in the animals as well as the feed as such. The next important aspect is that mixed micelle formation. The mixed micelle formation in the non-ruminants, if you take, right, the monoacyl glycerol play an important role in the formation of this mixed micelle. Whereas in the ruminants, the lysophosphatidylcholine play an important role in the formation of the mixed micelles. This is one important difference you need to understand in the mixed micelle formation of the ruminants and the non-ruminants. One is the lysophosphatidylcholine in the ruminants, whereas in the non-ruminants it is monoacyl glycerol. Then the role of protozoa in the dietary lipids or in the microbial digestion of the fats. So the protozoa usually absorbs some of the polyunsaturated fatty acids, and as it absorbs some of the polyunsaturated fatty acids, it will lock into the cell structure. As it locks, then it protects that polyunsaturated fatty acids from the hydrogenation process in the rumen. Otherwise, what will happen? The hydrogenation process will just disintegrates the polyunsaturated fatty acids, right? So that is why the hydrogenation process escape is very, very important, and the protozoa helping in this aspect. So this PUFA, which is integrated into the cell structure of the protozoa, which will undergo the intestinal digestion, which is acted upon by the enzymes as well as the acids in the stomach as well as the small intestine, and releases the PUFA, polyunsaturated fatty acids. And these polyunsaturated fatty acids released in the small intestine is the main source of the PUFA in the ruminants. Remember, otherwise what will happen? This hydrogenation process in the rumen itself will totally completes or utilizes all the PUFAs. So as a result, it may not be available for an animal. So that's how the protozoa is helping the animal, okay, by consuming lot of starch, and then on the exchange it is giving the polyunsaturated fatty acids in addition to some starch granules and then some microbial proteins to them. Let us summarize the total microbial digestion: carbohydrate, protein and fats, right? So the first is that cellulose and the starch, these are the carbohydrates, right? So the components cellulose and starch, majority, you can classify everything under these two categories, right? So the cellulose, where it will be acted upon by the cellulolytic bacteria, then starch, it is amylolytic bacteria, and ultimately it forms volatile fatty acids. Remember, volatile fatty acids. Now, next is that the non-protein nitrogen and the dietary protein. The dietary protein, again, it forms the volatile fatty acids, and then it liberates some ammonia, whereas non-protein nitrogen, it will get converted into ammonia, and then it forms the bacterial proteins. Now, this ammonia can be used for bacterial protein, or you can say along with the volatile fatty acids and the ammonia, it together forms the bacterial proteins, right? Now, the volatile fatty acids, from where and all it is formed? One is from the dietary protein, and next is that from the cellulose and the starch. Now, the starch, I told, it forms the metabolic acids. That means even from the protein also branched volatile fatty acids used to be formed, right? Then these volatile fatty acids, there are three volatile fatty acids. One is acetate and butyrate. These are the ketogenic in nature. The propionate is a glucogenic in nature. So as I told, the propionate is a glucogenic in nature. That means it will form the glucose, okay? And then the acetate and butyrate is a ketogenic in nature. As a result, it forms the fat, and then it can be used for the energy purpose. Then let us go to the bacterial proteins, where we left the high biological value bacterial protein synthesis. These bacterial proteins will get converted into amino acids or the peptides. Then these amino acids will be used for the production of the glucose, and then these amino acids is used as stored as a protein in the ruminants. And then sometimes the amino acids get converted into carbohydrates if you study the biochemistry different pathways. On the other hand, the amino acids can get converted into fats also, and then energy molecules. It can be used as an energy molecule. Then the oils, the fat substance, again it will form the branched volatile fatty acids that we have seen in the few slides before, and then it forms the saturated fats. The saturated fats is used either for energy production purpose, or it will go and settle in the ruminant body in the form of carcass fat, or it is a milk fat, right? And another important aspect is that even the acetate is very, very important for the production of the milk fat. It is having a direct impact on the fat production. Then the cobalt. Cobalt is used by the rumen microbes for the synthesis of vitamin B complex. So instead of giving a vitamin B12 injection or vitamin B complex injection to animal, it will be more effective if we feed or if we inject the cobalt to an animal, where the microbes will synthesize its own vitamin B complex in the ruminants. This is one important aspect. So we have seen the different diets, and then the fermentation process, what are its formed the intermediaries, and what is its fate in the ruminants. So this is the overview of the three digestions, or the microbial fermentation and the microbial digestion of fat, protein and carbohydrates, what we have seen in the last few classes. Then the next aspect is the chewing the gut, or you can say rumination. I told the ruminants used to spend around 8 hours per day just for chewing the gut or rumination process. This is nothing but the regurgitation or retrograde antiperistaltic movement of the intestine that leads to the chewing the gut. So the regurgitation brings by larger particles from the rumen to the mouth to produce the particle size as well as to increase the surface area so that it is available for the bacterial attachment. Now this is the most important aspect in the regurgitation. Why the ruminant is regurgitating or ruminating? It is just to reduce the particle size, because one important requirement I told, the particle size should be 2 millimeter in length, then only it will pass to the next compartment. So for that also this regurgitation process is required. And next is that even the surface area for these feed particles also should be increased so that the enzymes as well as the microbes will act on it, and it will break down into a simpler molecules of the any type of the carbohydrates or the proteins in the simpler way, right? The next is that this regurgitation is promoted by the presence of the rumen raft. Rumen raft means, I told, it is a fibrous material that is float on the rumen fluid. Now this fibrous material which is float on the rumen fluid, that causes some tactile sensation. Okay, this tactile sensation stimulates the acts on the ruminal epithelium. Then the rumen wall will cause a retrograde antiperistaltic movement. It causes, as a result, it will just bring the food particles from the stomach, that means the rumen, to the mouth, and then it will that go for remastication, and then it will redeglutition will takes place. The presence of the fibrous material, I told, that results into the tactile sensation of the epithelium of the rumen. As a result, it initiates the retrograde antiperistalsis contraction of esophagus, and that propel the cut into the mouth. So this is the mechanism by which it is bringing back into the mouth, and then it will chew it, and then that is mastication, remastication, you can say. It is not mastication. Then redeglutition will take place. Redeglutition means it is engulfing. Redeglutition means it is again after chewing, it will enter into the esophagus and it will go to the rumen. Now next, one important condition where I want to stress is that bloat. What is a bloat? Bloat is nothing but it is the accumulation of gas into the lumen, because it cannot be able to escape. I told there are different phases in the rumen digester, that is called a stratification, you can say, in the rumen digester. There are different stratas are there, different phases. So the topmost phase is the gaseous phase. That gaseous strata, you can say, that is, it is majority of the carbon dioxide and the methane gases you can find in there. So these gases must be belched out, or you can say it has to be removed from the rumen, right? Burping in humans, you can say. So if it cannot escape, then it will lead to the two types of bloat conditions in the ruminants. One is that dry bloat, another one is frothy or wet bloat. So the dry bloat means simply there is a blockage in the esophagus or in any tract. Because of that reason, it could not able to escape, leads to the dry bloat. If we remove or if we correct that measure, then immediately it will be removed. Then the animal will be normal. Whereas the frothy or wet bloat, in this condition, what happens is that the saponins will come and it forms the bubbles. Okay, that these bubbles causes the frothiness in the rumen, and it is very serious condition, and it has to be relieved immediately by the intervention, and the veterinarian knows how to relieve this frothy blood also. So okay, so this is about the bloat. That is one important condition. Of course, apart from that, there are many other conditions are associated with the rumen. Okay, that is another important ocean actually, the condition which is associated of the rumen. Then next we'll move on to the microbial digestion, the avian digestion. Okay, I told we are not going to touch all the aspects of the avian digestion. What are the important differences you need to know in related with the digestion? I'll stress on that. First, avian means birds, right? The teeth is absent for a bird, and instead of that, the lips and cheeks are getting replaced by the beak. Here you can say this is a beak, right? Then the stomach is two-chambered in the birds. We'll see what are those, right? One is the proventriculus, you can say the gizzard is there, right? Ventriculus and the gizzard. Then the ceca also is double, okay, in double nature, and the birds can produce around 7 to 30 ml of the saliva per day, right? And there is no epiglottis in birds. Then the taste buds are present. That means almost 300 taste buds are present in the birds. Even related with these taste buds, already we have seen in the neurophysiology, the special senses in the respiratory system. Then the salivary glands is well developed in the chickens as well as the turkeys. And coming to the esophagus is classified into two. One is the before the crop and after the crop. Okay, here this is the crop. One is the esophagus which is before the crop, that is called as pre-crop, and the esophagus which is after the crop, that is called as post-crop portion. Okay. Then what is this crop? I told now the crop is, right? What is this crop? This crop do not have any mucus secreting glands. Okay, so then what will be the function of this crop? The main function of the crop is to store the food. Okay, so the one and only function of the crop is to store the food. It do not secrete any type of mucus, and that is the importance of the crop. And the esophagus which is present above the crop is a pre-crop, and below the crop is the post-crop. Don't forget. Then next is the stomach of the birds. It has two stomach. One is glandular stomach. Gland means what? Gland used to secrete. So that is a glandular stomach, and another one is a muscular stomach. Muscular stomach. What for muscle is used? Muscle is used for the contraction purpose, right? The same way here also it is related with that. Then the glandular stomach, the two stomach I told you, one is proventriculus and another one is ventriculus, or you can say gizzard. So the proventriculus is a glandular stomach. The ventriculus or gizzard, you can say it is a muscular stomach. Okay. Now the stomach, next time when you go to the abattoir or the butcher shop, where they used to slaughter the birds and all, so you can see that the stomach color, this stomach color is red just because of the myoglobin in nature. Okay. So next will be the ventriculus. Ventriculus, I told it is a muscular stomach, or you can say the gizzard. Gizzard is here. This is lined with the mucosal surface that is linked with the cuticle or the chyolin. What is this chyolin is? Chyolin is a protein polysaccharide complex which lines the mucosal surface that is linked with them. Now, this cuticle or chyolin protects the ventriculus from acid, because this gizzard is present after the proventriculus. Proventriculus is having the glands, that means it secretes the acids as well as the other enzymes, right? So the content will come and act on the gizzard. So this gizzard is already having a coating of the cuticle or the chyolin. So that protects from acid as well as the proteolytic enzymatic action. Then this gizzard also contains another one important feature, is that grit or small stones or sand. This is used for grinding hardwoods, and then the microbial activity also observed in the crop, where the major fermentation products are lactic as well as the acetic acid. So if you see the ventriculus or the gizzard, two important aspects. One is the cuticle or chyolin, which is a protection purpose, it is there, and the next is the grit, which is required for the grinding of the hardwoods, and the microbial fermentation also happens in the grit. where the lactic and acetic acid is the major fermentation product. Then the avians are having 3 pancreatic ducts and 2 bile ducts, one for gallbladder and one for right lobe of the liver. And the antiperistalsis occur in the colon usually, the retrograde movement. This is required to move the urine from cloaca into the colon and ceca so that the maximum water reabsorption will take place, as well as for filling the ceca. Before the defecation, this antiperistalsis movement ceases, that means stops. Then the nitrogenous component of avian urine is that uric acid, which is less toxic compared with the ammonia. Then coming to the gastrin, it stimulates the acid secretion from the proventriculus. Next, coming to the avian vasoactive intestinal peptide, this is more potent than the avian secretin in stimulating the bicarbonate secretion. So let us see if it is a bicarbonate secretion, then the avian VIP is more important, vasoactive intestinal peptide, than the secretin. Then re-esterified triglycerides are packed into portomicrons in the avian, whereas in the mammals it is chylomicrons. So these many days you might have heard of the chylomicrons, but now in the avians you can say it is called as a portomicrons. The re-esterified triglycerides are packed as a portomicrons, not the chylomicrons, which is another important difference. Then birds do not have lacteals in the villi. As a result, the portomicrons is absorbed directly into the hepatic portal blood supply. Okay, there is no lymphatic system. Okay, so there is no lacteals in the villi. So as a result, the portomicrons has to be absorbed by the hepatic portal blood supply. Then the important volatile fatty acid is that acetate is the predominant volatile fatty acids in the ceca of avians. So if you see the alimentary tract of avian is that esophagus, pre-crop esophagus, it is divided, right? Then crop or ingluvius, and the post-crop esophagus, and then glandular stomach or proventriculus, then it will enter into the isthmus, then muscular stomach or gizzard, and then proximal duodenum it will enter, then liver, gallbladder, and the pancreas that will come and joins in between the proximal duodenum and the distal duodenum, right? And then the distal duodenum to jejunum to Meckel's diverticulum, it is a remnant of the yolk sac. Then ileocolic junction, then ceca, colon, bursa fabricius, cloaca, and when cloaca is a common opening, 3 different systems in the birds. One is the reproductive system, another one is urinary or the excretory system, then the digestive system, the cloaca, right? This is a common opening. So let me summarize what we have seen today is that microbial fermentation of the fats, that means microbial digestion of dietary lipids we have seen, and then the summary of carbohydrate, protein, and lipid microbial digestion. And at the end, we have seen what are the important aspects of the avian digestion. So with this, we complete the ruminant digestion, including the avian. Thank you.